So RBN-2397 is a first-in-class PARP7 inhibitor, which reactivates interferon signaling and anti-tumor immunity. By way of background, PARP7 is a stress-induced mono-ADP ribosyl transferase that is not typically expressed in normal tissues. And PARP7 expression actually blocks anti-tumor immunity and inhibits tumoral type 1 interferon signaling. And importantly, PARP7 is amplified and highly expressed in a subset of cancers. And so that really provided the rationale to develop RBN-2397, which is an oral, potent, selective small molecule inhibitor of PARP7. And in preclinical studies showed very impressive complete tumor regressions in preclinical models as a single agent and also in combination with anti-PD-1 therapy. And so on this clinical trial, which was a phase 1 study looking at RBN-2397, this was found to be biologically active and with a favorable safety profile. So RBN-2397 overall was well tolerated, with the majority of toxicities being mild to moderate. Importantly, there were no significant chronic or immune-related toxicities observed, and there was preliminary anti-tumor activity observed in both the dose escalation and dose expansion cohorts. For example, we observed a partial response by RECIST criteria in an HR-positive breast cancer patient during dose escalation, and a partial response by RECIST criteria that was very durable in a head and neck squamous cell cancer patient. We also saw multiple other patients achieving RECIST stable disease that was durable. And importantly, we found that PARP7 expression was seen in all evaluated baseline tumor biopsies with a high expression in tumor cells versus stroma. Induction of adaptive immunity as well following RBN-2397 was evidenced by increases in CD8 positive T cells and granzyme B expression in most expansion cohort patients across different cancer types. And increased expression of various immune checkpoint proteins, including PD-1 on T cells and PD-L1 on tumor cells, were also noted in a subset of samples which underwent analysis. And so overall, these data validate PARP7 as a novel anti-cancer therapeutic target and confirm proof of mechanism of RBN-2397. And these findings really support the ongoing trials of RBN-2397 in combination with pembrolizumab, and also another trial in combination with nivolumab.